Hi students, good morning to everybody. I have to continue the lesson, chapter 6, Respiration. What are the contents? The remaining parts of the lesson. Now we have to know something about the respiratory pigments, those pigments which are involved in the transport of gases. Now we are talking about only one pigment mainly in human beings, that is in vertebrates, hemoglobin. So it is a conjugated protein because it is being formed of a protein part plus a non-protein part. Now the protein part is globin, this is a non-protein part. That's why it's called as a conjugated protein. So it is a conjugated protein formed of two components. One is protein, another one non-protein. For example, hemoglobin, it is formed of heme, a colored pigmented part, and a colorless what is called protein, globin. Now, if you are taking the heme, what is heme? It is an iron containing pigmented portion. Iron containing pigmented portion, which constitute normally about 4% of what is called hemoglobin molecule. So in each hemoglobin molecule is being formed of an iron containing pigmented portion constituting 4%. The next part is the globin. It is nothing but a colorless protein. A colorless protein which belongs to one class, what is called histone class. We have different types of proteins, different classes of proteins. One type of protein is a basic protein, what is called histone. So it is a colorless protein, that is a globin. Normally it is being formed of 4 polypeptide chains, 2 alpha chains and 2 beta chains. It is not given in the above. Now the molecular weight of each molecular hemoglobin is about what we have 68,000 Daltons. This is the molecular weight. Now if you are taking one hemoglobin molecule, particularly the heme, it contains 4 iron atoms. 4 iron atoms. Each iron atom is capable of carrying 1 molecule of oxygen. So the oxygen carrying capacity of one hemoglobin molecule is 4 molecules of oxygen. So a molecular hemoglobin carries 4 molecules of oxygen. In terms of what we have, actually the number of molecules of oxygen. Okay, so it contains 4 atoms of iron, and each iron atom is capable of just normally carrying 1 oxygen molecule. That's why each hemoglobin carries 4 oxygen molecules. Now this is the main hemoglobin. Sometimes we have less than 1% of hemoglobin is found in the form of abnormal hemoglobin. This abnormal hemoglobin is called methemoglobin. So normally the iron present in the heme part is in ferrous state in normal hemoglobin. If that iron is in ferric state, then it's called methemoglobin. This is because of oxidation. Normally hemoglobin is oxygenated, that is nothing but carrying of oxygen. Then when you have the oxidation of what is called the iron atom from ferrous to ferric state, then the hemoglobin is called methemoglobin. Normally methemoglobin is caused because of pollutants in water. Sometimes pollutants in water that causes the formation of methemoglobin. That leads to what is called some syndrome, what is called blue baby syndrome. The affected child becomes actually having a blue tint, blue color of the skin. That's called blue baby syndrome. So if the concentration of methemoglobin increases, we have a disorder in the case of child, that is what's called blue baby. Okay, so when the iron atom is in ferric state, then what we have the ferrous state, it is called methemoglobin. So this methemoglobin does not bind with oxygen, has no capacity of binding with oxygen when the iron atom is in ferric state. So normally RBC contains less than 1% of methemoglobin. If the percentage increases, then only we have, that is what we have, the blue baby syndrome. Particularly in the case of children. This is because of pollutants. Sometimes arsenic in water may cause that one. That's about the respiratory pigments. Main pigment hemoglobin, the abnormal pigment methemoglobin doesn't take part in the transport of oxygen. Now the next one, the transport of gases. The next one, how oxygen and carbon dioxide transported oxygen from the alveolus to the tissues and carbon dioxide from the tissues to the alveolus through the blood. Now first one, the transport of oxygen. So molecular oxygen is carried in the blood by two ways. Number one, in dissolved form, in plasma. Least percentage of oxygen is actually transported in dissolved form, in plasma. And another one, bound with hemoglobin within RBCs. RBCs contain the respiratory pigment. It is having affinity with oxygen. It binds with oxygen to form oxyhemoglobin. So the second way of transport, bound with hemoglobin within RBCs. Now let's take the first one, transport in dissolved form. Now oxygen is poorly soluble in water, so only 3% of oxygen alone transported in dissolved form, as it is having less solubility, only 3%. Now the second one, 97% of oxygen binds with hemoglobin in a reversible manner to form oxyhemoglobin. So here, hemoglobin binds with oxygen to form oxyhemoglobin, that is happening in the alveolus, and it dissociates in the tissues once again to form hemoglobin and oxygen. So it is a reversible reaction. Okay. Now, the rate at which the hemoglobin binds with oxygen depends on the partial pressure of oxygen. More is the partial pressure of oxygen, as in the case of alveolus, it binds readily with oxygen. Less in partial pressure in the tissues, it normally dissociates. The binding force, the affinity is being reduced. That is what we have. So each hemoglobin contains 4 atoms. So the maximum number of oxygen molecules carried by one molecule of hemoglobin is only about 4 molecules of oxygen. This is oxygen carrying capacity of hemoglobin in terms of molecules. So each hemoglobin carries a maximum 4 molecules of oxygen. Okay, we'll continue further about actual transport of oxygen. The factors favoring the formation of oxyhemoglobin in the alveolus and the factors favoring the dissociation of oxyhemoglobin to oxygen at the tissues or in the tissues. Now what are the factors favoring the formation of oxyhemoglobin and its dissociation? Now the formation of oxyhemoglobin occurs in the alveoli. The dissociation of oxyhemoglobin occurs in the tissues. Now let's see, what are the factors favoring the formation of oxyhemoglobin in the alveoli? There are 4 major factors. Number 1, in the lungs we have high partial pressure of oxygen. When compared to high partial pressure of oxygen, the partial pressure of carbon dioxide is low. The temperature is also low. Less hydrogen ion concentration. When you have less hydrogen ion concentration, the pH is normally more. Alkaline pH. We can say alkaline pH. So 4 factors, high partial pressure of oxygen. The remaining 3 factors are less. Low PCO2, low temperature and less hydrogen ion concentration. These factors favor normally the formation of oxyhemoglobin. Now in the tissues, what is happening? The dissociation of oxyhemoglobin to oxygen occurs. Now these are the factors. Now what are the factors favoring the dissociation of oxyhemoglobin to oxygen in the tissues? So the reverse is the case about these factors in the tissues, where we have low partial pressure of oxygen, then high carbon dioxide concentration or high partial pressure of carbon dioxide. We can say in terms of partial pressure of carbon dioxide, high PCO2. So when compared to the lungs, in the tissues we have the partial pressure of carbon dioxide is high. Then high temperature. All are just in the reverse order here. When compared to what we have the alveolar, there is medium. High temperature, high hydrogen ion concentration. When the concentration of hydrogen ion increases in a medium, in a fluid medium, now we have the acidity increases. When the acidity increases, the pH becomes low. So that's why I mentioned about low pH. So increase in hydrogen concentration increases acidity and decreases the pH. And here decreases in hydrogen and concentration increases the pH, that is alkaline pH. So these are all the different factors favoring the formation of oxyhemoglobin and its dissociation. Okay, let's go for the next one, oxyhemoglobin dissociation curve. What is oxyhemoglobin dissociation curve? A sigmoid or S-shaped curve has been obtained when percentage saturation of hemoglobin is plotted against the partial pressure of oxygen. So we are taking two units and they are being plotted in a graph. One is the percentage saturation of hemoglobin with oxygen and the second one partial pressure of oxygen. Now what is percentage saturation of hemoglobin? This is a percentage of hemoglobin just normally binds with oxygen. Percentage of hemoglobin binds with oxygen. In a normal condition, the hemoglobin, what we have, the percentage of hemoglobin binds with oxygen or percentage saturation is about 97%. We cannot receive 100% saturation. We can receive only 97% actually saturation only. That's why we have, that is when you have 104 millimeter mercury of oxygen, that is in alveolar air. We have only 95 millimeter mercury in the oxygenated blood. You see the difference. That's why I said there is no 100% saturation, only 97% saturation of hemoglobin with oxygen. So, a sigmoid curve is obtained when percentage saturation of hemoglobin, it is nothing but percentage of hemoglobin binds with oxygen, is plotted against the partial pressure of oxygen. And this curve is called oxyhemoglobin dissociation curve. The curve is normally S-shaped or sigmoid in shape. Okay. So, from this curve, one can find out the effect of partial pressure of oxygen, partial pressure of carbon dioxide, temperature, hydrogen and concentration, etc. The result is being drawn to find out the effect of these factors. Okay, this S-shaped curve has a steep slope. You see that one, there is a steep slope between 0 and 50. And then it becomes flattened. So it becomes flattened. Then after you see that one 70 to 100. After 70, between 70 to 100. So this S-shaped curve has a steep slope for partial pressure values between 10 and 50 millimeter mercury, and then flattens between 70 to 100 millimeter mercury. You can see that one, the flattened nature. This is the steep slope between 10 and 50. This is the flattened nature of the curve. Okay, under normal physiological conditions, 100 ml of oxygenated blood delivers only 5 ml of oxygen. If you are taking 100 ml of blood, it contains 20 ml of oxygen. Not all the 20 ml of oxygen is delivered to the tissues. Only 5 ml of oxygen out of 20 ml of oxygen in 100 ml of blood alone delivered. The remaining is present as a reserve in the blood itself. It's being retained as a reserve in the blood itself. So, only 5 ml of oxygen to the tissues is delivered in 100 ml of blood of oxygen. So, 100 ml of oxygenated blood, we can say. 100 ml of oxygen. So that's about oxyhemoglobin dissociation curve, and also the transport of oxygen is over. We have to just skip to the next one, the transport of carbon dioxide. Now, how is carbon dioxide transported from the tissues to the lungs or alveoli? Now, normally blood transports carbon dioxide from the tissues to the lungs in 3 different forms. Number 1, dissolved in plasma. About 7 to 10% of carbon dioxide is transported in dissolved form in plasma. It combines with water to form carbonic acid that is being transported in dissolved form, only least amount. The second one, bound to hemoglobin. About 20 to 25% of dissolved carbon dioxide is bound and carried in RBCs, that is as carbaminohemoglobin. That is, the hemoglobin binds with the carbon dioxide. We have what is called the amino group present in the protein of globin, which combines with the carbon dioxide, so that we have received what is called carbaminohemoglobin. Okay, so we have nearly 586 amino acids in hemoglobin. Each amino acid has an amino group and a carboxyl group. Now the carbon dioxide combines with the amino group to form a carbaminohemoglobin. Simply the equation is represented as hemoglobin plus carbon dioxide, carbaminohemoglobin. But the major amount of carbon dioxide is transported in the form of bicarbonate ions, as bicarbonate ions in plasma. about 70% of carbon dioxide is transported as bicarbonate ions. Okay, so this transport is influenced by the partial pressure of carbon dioxide and the degree of what we have hemoglobin oxygenation. Depends on the partial pressure of carbon dioxide and the degree of what we have hemoglobin oxygenation. So the reaction, the formation of what we have actually carbon dioxide with what is called actually, you see that one water to form carbonic acid is facilitated by an enzyme what's called carbonic anhydrase. Now this carbonic anhydrase is nothing but an enzyme present in higher concentration in the RBCs. It is also found in little concentration or small concentration in the plasma. This is a major enzyme responsible for the formation of carbonic acid and its dissociation. Suppose in the animal kingdom, we have, this is the fastest enzyme. This is the fastest enzyme in the animal kingdom. In the presence of carbonic anhydrase, we have received at least 300 what is called actually, that's carbonic acid molecules within a second. But without this one, it takes more time. So it facilitates the formation of what is called carbonic acid and its dissociation in the reverse direction. Both the directions it is working to form carbonic acid as well as its dissociation to form hydrogen ions and bicarbonate ions. I told you, I just mentioned the equation also. Carbon dioxide is transported in 3 different ways by the blood. Now what is the mechanism of transport of carbon dioxide? At the tissues, the partial pressure of carbon dioxide is high due to catabolism. Because of the enzymatic oxidation of the food, the glucose carbon dioxide is released. Carbon dioxide diffuses into the blood to form bicarbonate ions and hydrogen ions. Then what's happening? When carbon dioxide diffuses the RBC, now it first enters into the blood to form carbonic acid, which it dissociates to form, that is what we have bicarbonate and hydrogen ions. The same manner, from the plasma, carbon dioxide enters into the RBC, where it combines with water present in the cytoplasm to form carbonic acid. This reaction is catalyzed by the enzyme what I mentioned, carbonic anhydrase. This is the enzyme, formation of carbonic acid. The carbonic acid is a weak acid and it is unstable. So it immediately dissociates to form what is called hydrogen ions and bicarbonate ions. Hydrogen and bicarbonate ions. This reaction is also catalyzed by the carbonic anhydrase. The formation of carbonic acid by what is called carbonic anhydrase, its dissociation to form hydrogen and bicarbonate ions, it is also facilitated by what we have the same enzyme. You can see the reaction. Now the carbonic anhydrase facilitates what we have the reaction in both the directions. That is what we have carbon dioxide combines with water to form carbonic acid, either in the plasma or in the RBC. Because the enzyme is present in both the places, but the concentration is high in the RBCs. Now this carbonic anhydrase facilitates the formation of carbonic acid and also the splitting of carbonic acid to form carbon dioxide and water. The same manner, the carbonic acid is dissociated to form bicarbonate ions and hydrogen ions. Here dissociation, here is the splitting. The reaction is also a reversible one, catalyzed by the enzyme carbonic anhydrase. Okay, then what will happen? Now the carbon dioxide actually enters in the blood, combines with water forming carbonic acid, dissociation to form bicarbonate ions and hydrogen ions. Now the bicarbonate ions from the RBC quickly moves out of RBC into the blood or into the plasma, from where it is transported to the lungs. Now in the lungs, now the reverse reaction occurs. What's happening? The bicarbonate ions combine with what's called hydrogen ions to form carbonic acid, which splits into carbon dioxide and water. So carbon dioxide is trapped as bicarbonates in the tissues. It is transported to the lungs and eliminated as carbon dioxide. Now every 100 ml of deoxygenated blood, every 100 ml of deoxygenated blood delivers only 4 ml of carbon dioxide to the alveolus for elimination. I mentioned about 5 ml of oxygen alone delivered by the oxyhemoglobin to the tissues, that is in every 100 ml. Here in every 100 ml, that is in deoxygenated blood, only 4 ml of carbon dioxide alone just released into the alveolus from where it is being released outside. I am just write down on the board about that one. So anyway, bicarbonates from just actually RBC diffuses into the plasma and carried to the lungs. In the lungs or in the alveoli, the reaction is reversed. What will happen? The bicarbonates normally combines with the hydrogen ions to form carbonic acid, which is splits into carbonate and water, and carbon dioxide is eliminated. So, carbon dioxide is trapped as bicarbonates in the tissues and is transported to the alveoli for its elimination as carbon dioxide. And 4 ml of carbon dioxide alone delivered to the alveolus for elimination by the deoxygenated blood. I will draw a simple diagram also to show how far actually the transport of carbon dioxide occurs. Now the bicarbonate ions formed inside the RBC after the dissociation of carbonic acid moves quickly from the RBCs into the plasma where it is carried to the lungs. So the hydrogen ion is retained inside the RBC, it combines with the hemoglobin, so that we have formed reduced hemoglobin. We don't want that one. So the bicarbonate ions, you see that one that is formed here, this is the RBC, moves out of RBC and transported as bicarbonate ions to the lungs in the form of either sodium bicarbonate or potassium bicarbonate. Now, actually, at the alveolar side, where the partial pressure of carbon dioxide is low, the reaction is reversed. Now, what is happening? That is, we have the hydrogen ions and the bicarbonate ions combine together to form carbonic acid. Now the medium or the environment, the alveolus, favors the splitting of what we have carbonic acid to form carbon dioxide and water. Now carbon dioxide is eliminated. So the carbon dioxide is trapped as bicarbonate ions at the tissue level. It is transported to the alveoli for elimination as carbon dioxide. Every 100 ml of, you see that one deoxygenated blood delivers only 4 ml of carbon dioxide for elimination. Now here is a simple diagram and showing that one how far actually the transport of carbon dioxide occurs. Now carbon dioxide is formed inside the cells as a result of catabolism. It enters into the plasma of the blood, from where it enters into the RBC, where it combines with water to form carbonic acid. This reaction is characterized by the enzyme carbonic anhydrase. It is unstable because it is a weak acid. It dissociates from hydrogen ions and bicarbonates. Now the bicarbonates diffuse out of what is called actually the RBC, and it is transported as bicarbonate ions in the form of either sodium bicarbonate or that is potassium bicarbonate. Now in the lungs or alveoli, the reaction is reversed. The hydrogen ions combine with the bicarbonates to form to form carbonic acid, which is splits into carbon dioxide and water, and carbon dioxide is eliminated. That's the thing what is happening is simple, just actual diagram showing how far the formation of carbonic acid and its dissociation occurs with the help of carbonic anhydrase. So with that I concluded about what we have, the transport of carbon dioxide. Now let's see the regulation of respiration. Now we have a respiratory rhythm center, a specialized respiratory center present in the medulla oblongata, commonly called as the respiratory center, just actually in the hindbrain. It is called respiratory rhythm center. This is the main center controlling the process of respiration. Okay, so this is the one which is involved in the regulation of respiration, medulla oblongata of hindbrain. Now we have in the respiratory rhythm center, we have expiratory center and inspiratory center, just actually to control inspiration and expiration. There is a center which regulates both respiration, what we have, and inspiration. So inspiration and expiration, they are being actually modified or moderated by another center what's called pneumotoxic center. Pneumotoxic center, not toxic. Pneumotoxic center. It is present normally in the pons region, normally called as the pons varoli of brain, which moderates the function of the respiratory center. Both inspiratory center and expiratory center, they are present together as respiratory rhythm center and their functions are moderated by what we have this pneumotaxic. So pneumotoxic center. So it is present the pons varoli and which moderates the function of the respiratory rhythm center. Coordination. For example, when the expiration is there, there is no inspiration. When the inspiration is there, the inspiration actually is carried to the inspiratory center, which then sends impulses to this center, what's called pneumotaxic center, which stops inspiration and stimulates the expiration process. That's why I said it modifies the function of respiratory rhythm center to ensure effective breathing. Now, chemosensitive area. An area which is present near to the what is called respiratory rhythm center, which is sensitive to carbon dioxide and hydrogen ions. These also, what we have, these respiratory rhythm centers like that because some receptors are found, that is in what we have aortic arch and the carotid arteries, the one which supplies blood to the brain. Because certain receptors present in the aortic arch, that is what we have in humans, we have left aortic arch. And the carotid arteries, they send sensory signals to the rhythm center. They are just stimulated according to the concentration of hydrogen and the carbon dioxide and not oxygen. So they send actually stimuli or the signals to the respiratory rhythm center, which immediately act for remediation. According to the concentration of carbon dioxide and hydrogen ion, we have either inspiration or expiration. That is the thing what is happening. That is called remediation. Stimulate the remediation process of what is called respiratory rhythm center. Now, the role of oxygen is insignificant in the regulation of what is called respiration. There is no role played by oxygen. Now, the respiratory center is not stimulated by the concentration of oxygen, or we can say partial pressure of oxygen. That's why I said the role of oxygen is insignificant. There is no role played by what is called oxygen in the regulation of respiratory rhythm. That is what we have inspiration and expiration. Mainly carbon dioxide and hydrogen ions are responsible for regulating the respiration process. Okay, so we have respiratory rhythm center, pneumotaxic center, the one which modifies what we have actually the breathing process, and chemosensitive area present nearer to what we have the respiratory rhythm center, and also we have the receptors present in the aortic arch and the carotid arteries, the one which supplies blood to the brain. And they only just send signals to what is called actually respiratory rhythm center to regulate the process of breathing. Now, oxygen has no role in the process of what is called respiration.